I'm going to very specifically talk about taxonomy and the importance of taxonomy for us in the room here today. I think it would have been too much if I'd also had to talk about ecology and biology, but this is just as a way to show you that there are many things that are important when we study, in this case, viruses and virus surveillance, and there are many things we don't think about, or we don't think that they're important, or we simply don't understand. Taxonomy is one of those things, and I would like to try and get the message across today that this is really not just important, it's actually crucial. I believe that most of the people in the room are not zoologists as such. Many of you are not even biologists. I think you come from a medical background, so I hope that this will be at least interesting. So bats are mammals, but they're not just mammals. If you look at, this is a pie chart showing the number of species of different types of mammals, and you can see that rodents make up about 40% of all mammals. Bats make up another 20%. So bats and rodents together make up more than half of all mammal species. And if you include shrews, then we're getting close to 3/4 of mammals comprising small mammals. So if you want to, if you say, when a normal person thinks about mammals, they think about large mammals, but yet the diversity of mammals is actually in the small groups. And by the way, if you want an updated mammal list and mammal taxonomy for the world, take note of that website. It is very much up to date, and it's live. It's updated as they go along, and their taxonomy is very good. So we're talking about bats in this conference, and bats are also my favorite group of mammals. They are wonderful things. These are just a very small number of the species I've encountered and that I've captured and photographed across the continent. And as you can see, they are absolutely gorgeous. So if you're not yet bat enthusiasts, hopefully you will become bat enthusiasts, because really these are not carriers of disease. These are interesting and beautiful animals in their own right. More than 1,400 species of bats have been described in the world. And if you want to know about bats and the absolute latest taxonomy and naming of bats, please refer to this website here. They are very good at keeping things up to date, and that list grows. I had to change the number here. I gave this slide, this particular, not this talk, but the slide I gave recently, a month ago, 2 months ago. I had to go and change that number by about 30 species in just 2 months. Now, bats are diverse, second most diverse group of mammals, yet they are highly understudied. And this is just an illustration of how understudied they are. This is data that comes from the Bible of African Mammalogy, the 6-volume Kingdom's Mammals of Africa. If you look in those 6 volumes of the Mammals of Africa and you pull out how many papers have been published per species of mammal, this is what you get. For a typical primate, there are about 36, 37 papers published on that one species. Ungulates, which are the antelopes, similar numbers, similar for carnivores. Yet, if you look at bats, they are, for the typical species of bat, if the average species of bat, there are less than 5 papers published for that. And in fact, this is not very good. This is not a very, averaging this number is not very good, because for the vast majority of bats, there is only the original description of the species and nothing else. So there is very little effort that's gone into bats. So as a result, there is very little that we know about bats, especially on the African continent. And this leads us to all sorts of problems. Problems, in this case, of identification. Here are two species of bats that are photographed from the same location in Liberia. These may not appear to be, but these are two separate species. They are good species. They are different in genetics. They're different in echolocation calls. They're even different in ecology. Yet there is absolutely no way to distinguish these two species visually on external morphology. But to make things even more complicated, neither of these two species has a name. And so here we are dealing with the sort of challenges that the average person deals with when they're working with bats in Africa. What are you going to do if you're a virologist and you want to study diseases? What are you going to do in this particular case? So the point here is that knowing the species affiliation is actually very important. We've seen in the earlier talks how certain viruses or groups of viruses are only found in one species or in one genus or in one family of bats. We need to know what these species are if we're going to make sense of the viruses and the risk to humans. So let me take a step back now and ask the question, what is taxonomy? I'm sorry for those of you who know. Hopefully that's not all of you in the room. But for those who don't know, taxonomy really is just simply categorization and naming of organisms, living creatures. And the typical categories that we talk about, they go from the very broad, the domain, and kingdom, phylum, all the way down to genus and species. But where do we get these names from? Where do we get these categories from? They come from the phylogeny that, or the tree of life, where we relate things to each other based on their relatedness. Birds are most closely related to crocodiles, and that group is most closely related to snakes and lizards, then to mammals, then to amphibians, and more distantly to the lungfish. So we need a phylogeny before we can give an animal a proper name, or at least categorize it according to those categories that I mentioned. And where do we get these phylogenies from? Well, we've got several different ways in which we can, and we have in the past, created phylogenies. Originally, the traditional way of identifying relationships between animal groups was through morphology. So, for example, on the left there, you can see there's a series of skulls. The names of those are totally irrelevant, but you can see that even as a non-expert, you can see that there are very big differences in the shape of those skulls, with the very domed skull at the top there, whereas at the bottom skull, it's much more flat. The size of the teeth are different. Those are all bats, but they're morphologically very different in shape of the skull. And those shapes can be described. We can also use other features. It's not just the skull that we need to use. We can also use in this case, the shape of the wing. Is the wing long and narrow? Is it shorter and broader? And this is just as an example of one feature that we can use to describe the morphology of animals. Linked to morphology is the field of morphometrics. Here, we are not just describing the morphology, we are actually quantifying it. So we take that same skull that we looked in the previous slide. Here's my pointer. We take the skull and we measure different aspects of that skull. So for example, GLS, the greatest length of the skull. We can measure how long is that skull. We can measure how broad is it. How long is the tooth row? What's the distance between one tooth and the other tooth? We can measure large numbers of different characters. We can do the same for any other feature of the animal, such as the wing on the right-hand side. And then we can use fancy multivariate statistics to put all this together, in this case to show that we have two different species, one in the blue and another species, one in the red. Each of those dots refers to a different individual that has been measured, and you can see that these two species do not overlap in a morphometric space. And so it's not just about describing the shape of the skull, it's actually quantifying it and mathematically showing that there are differences. And then, of course, in the modern world that we live in, we can describe relationships based on genetics. We can sequence genes. We can sequence the entire genome even. And we can then show that genetically there are differences. This is the mitochondrial genome and here I'm just showing this to show that two very commonly used genes for mammals anyway, for bats, it's cytochrome B gene and the CO1 gene. Both of these genes work very well for most bats, for most mammals, and can be used to discriminate the different species. Now, what we do as modern taxonomists is to put all of this together. We take the morphology and morphometrics, we combine it with molecular biology, which is the genetics, and then we throw in anything else that we can find, the list down there. And where these overlap, this is what we call integrative taxonomy. So we don't take only morphology or only morphometrics or only genetics. We really try to combine everything and come up with a solid taxonomy that speaks to all of those. Now, in order for us to be able to do this, we need good reference collections. We need specimens, the actual individual animals that we can look at for the morphology, for the morphometrics. We need tissue samples, we need genetic references, and this brings me to the importance of vouchers and taking voucher specimens. It is absolutely essential for any study to be collecting these, especially in a place like Africa, where we have so many undescribed species and so much confusion in taxonomy. So taking specimens does not mean going to the field and killing everything that you can find. You are using your discretion to take one, maybe two specimens that you think will represent the species that you're dealing with, and putting them into an established museum. This is a photograph of a museum. I actually don't know which museum this is, but this is what a modern museum looks like. If you go to, I was recently, well, before COVID, I was at the Field Museum in Chicago for a sabbatical, and this is what it looks like. Row after row of drawers with hundreds of specimens in each drawer. We're talking hundreds of thousands of mammal specimens. This is unfortunately not the case in most of Africa. If we exclude South Africa, most of our museums look more like this. This is our national museum at the University of Eswatini. There is no funding for this, and it's very difficult to maintain the collection, but we do our best, and we've got, despite what the photos show, we've got a very important reference collection for the country, as well as important collections from other parts of Africa. So, associated with my talk, or an integral part of my talk, is actually the importance of museums for our work, because taxonomy is totally reliant on museums. Well, it's totally reliant on specimens, voucher specimens. Those voucher specimens are housed in museums. Hence, museums are vitally important to our role, our task. And museums are not well financed and resourced in Africa. So I want to give you an example now, or a couple of examples actually, of to show you exactly how this is all very important. How taxonomy, why I'm saying taxonomy is important for in this case, for virology. The first example comes from a species of bat called Hipposiderus lamotti. This bat was described, the picture I'm showing here is the original description by somebody called André Brosset, who passed away a few years ago. He published this description in 1984. And why I'm saying this will become important in a moment. Now, this bat was described from Mount Nimba, which is in West Africa. It's in Liberia / Guinea / Ivory Coast. The mountain cuts across those three countries. And I've shown this on top of a map of Africa there that shows species richness of bats in Africa. The orange colors, the deeper the orange, the more species there are. The more blue the color, the fewer species there are. And what I want you to note is that Mount Nimba is sitting actually right in the zone of high species richness. This is what Mount Nimba looks like. You can just, you can't actually see it well on the screen here, but Mount Nimba rising in the background with tropical rainforest in the foreground. And the reason Mount Nimba is so important is because of iron ore extraction. This is the pit that was created in the 70s and 80s, exporting iron ore, which you can see on the left there, over here. This is very high grade iron ore that can be shipped directly to the steelworks factory without any initial processing. So this is very much in demand, and as a result, mining continues on this mountain. The reason why Hipposiderus lamotti now becomes important is that Hipposiderus lamotti is only known from this one mountain, Mount Nimba, 30 kilometers from one end of the mountain to the other, a very small little mountain. And I was called up to verify whether this bat occurs on in the mining site where they're going to mine for the bat, where they're going to mine for iron ore. And the consultant that had been called up there was actually a bat specialist who had been called up by the zoologist to identify whether this bat occurs there or not. Nobody could identify whether this bat was there or not. They had caught a whole lot of bats, but they were unable to identify them because nothing was matching. So they called me in as a bat specialist to try and make sense out of all of this. So what I did is I created I sampled widely and collected bats from across the mountain from the genus Hipposiderus and analyzed it genetically, produced a phylogeny. And all you need to look at are these blue bars, which refer to the species of Hipposiderus that I collected from the mountain. Now, all of these blue bars could get names, but what was not clear was whether this bar over here was referring to Hipposiderus lamotti. The reason was that the original Hipposiderus lamotti had never been sequenced, so it was not clear what the relationship was. We know that these bats echolocate at different frequencies. So I recorded the echolocation of what we were calling Hipposiderus lamotti, and it was calling at 120 kilohertz, very different from the other Hipposiderus on the mountain. But the original Hipposiderus lamotti had never been recorded, so we had nothing to compare this with. Now the problem was here. The problem lay in the fact that the specimens that we collected did not correspond. The measurements did not correspond with the original Hipposiderus lamotti. And this is what I mean. If you take the original description by André Brosset and you compare these these morphological characters, he got 55.6 for the holotype, the specimen on which the bat was described. My 6 species were 55.9. Sorry, my 6 specimens were 55.9. No problem. That's almost identical. The same for skull length, zygomatic arch, but I draw your attention to the bottom 2 here. The first phalange is the first finger of this, and you can see the big difference here between 14 and 18, 12 and 20. And this was what the problem was, that our measurements simply did not correspond. So I was, by chance, at the museum in Paris. That's the Natural History Museum in Paris, and I was working with Christian Denis on, in fact, another book, The Rodents of Africa. And so I used that opportunity to go to the collections and examine the type specimen on which the bat had been described, which was housed in that, despite the Natural History Museum looking grand and beautiful in the big picture, the collections are actually in a really terrible looking room. It almost looked, in fact, I thought I had gone into the wrong place when I entered for the first time. It was more like a bomb, been hit by bombs and things. There's that type specimen. That's the specimen on which André Brosset described Hipposiderus lamotti. And still looking good after 40 years of preservation. And then I remeasured that specimen. Now notice that's the original measurement by Brosset, in brackets. That is my measurement of that exact same specimen. And then these are the specimens that I collected in the field. Can you notice that most of my measurements are identical in terms of those sort of finicky 0.1, 2, 3 millimeters difference is nothing. But look down here. Look at these 2 measurements. So Brosset had made a mistake in his measurements, and the only way we could verify this is because he had collected a specimen which was in a museum, which allowed me to go and visit and remeasure. And this whole confusion over lamotti was over the fact that he had mismeasured the original specimen. So again, showing how important it is to take voucher specimens and to make collections, because we do make mistakes, we need things to compare against. So this is another example from a different branch of the study of bats, and it has to do not with confusion over names and confusion over mismeasurement of things, but rather just the sheer diversity of unknown life in Africa. This is based on a paper by Fisher et al. It's published quite a few years ago now, 4 years ago, where they tried to estimate how many unknown species, undescribed species are there in the world of mammals. And I wouldn't pay too much attention to the numbers that they give here, But the important thing is that they ranked the Afrotropics as the highest in unknown species of mammals. They listed something like 120. They predicted that there are something like 120 species of mammals yet to be described. I would put that figure more like 300, but that's beside the point. Compared to the other continents, Africa by far has the greatest predicted unknown numbers of species. This means that we're really dealing in an area that requires a lot more input in terms of description of species. And so I want to talk a little bit about that and my own experience in this world. Going back to Mount Nimba, where the Hipposidrus lamoti story was, here are 4 species of vespers in the family Vespertilionidae that we heard of earlier this morning, that are important for certain types of coronaviruses. These are 4 species that I described that were unknown to science. None of these species are splits in subspecies or playing around with words or anything like that. All 4 of these species were unknown to science, and they were described as completely new species. Now, the irony here is when Brosset did his surveys and published in 1984, this is taken word for word from his paper. And for those of you who don't read French, it says, "The bats of Mount Nimba are among the best studied in Africa." So, and actually he was right, because by the time he went there, he was the fourth zoologist to go to Mount Nimba to study bats at Mount Nimba. And yet, despite the fact that it's been well surveyed, here are 4 species, and relatively common species, by the way, that I found several decades later. So can you imagine what you're going to find if you go to a very poorly known part of Africa? But it's not just in some faraway place in tropical Africa that you get new species. We are describing new species all over the continent. Here are just some examples from southern Africa. Rhinolophus gorongosi. This was recently described from Mozambique. In fact, all 3 of these are described from Mozambique. And I'll come back to the 2 Miniopterus in a moment over here. So new species are being described literally across the continent, regardless of whether they've been well visited by scientists or not. This continent is still very much in the exploration stages when it comes to bats. So now I want to look at a slightly different angle. Taxonomy, yes, it's about describing, naming species, giving species names, and those names help us in other fields. But it's much more than just giving names. It's also giving the relationships of other levels of the taxonomic hierarchy. So here's a slide showing different species in the family Vespertilionidae. And all of these bats here have jumped from one genus to another. One year they are Pipistrellus, and then they are Neromycia, and then it's Leiphotus, and then it is this, and it is that. And this is another thing that drives non-taxonomists crazy. In fact, it also drives taxonomists crazy. It's not just everybody is annoyed when a species name is changed, because it means you have to learn one more name. But the problem is that these relationships are absolutely vital, because if you put something in the wrong, if you give it a wrong genus name, then, and its relationships are wrong, you can end up with all sorts of misinterpretations, because if a virus is associated with a genus, very closely associated with a genus, but you have described the genus incorrectly, then you're going to look at your data and say, there's no relationship between the virus and the genus. And it's everywhere. But yet it's because your taxonomy was incorrect. So I want to really just end off by showing you some work. This was work I did on my sabbatical, looking at the generic relationships within one group of vespers, the pipistrelle-like bats. It's actually the vespers, by the way, the family Vespertilionidae is the largest family of bats. So bats are the second largest group of mammals. The vespers are the largest group of bats. And the pipistrelle-like bats are the largest group of the vespers. And what I want to show here is that, so these names are the genera of pipistrelle-like bats that are found in Africa. And what I want to show is that 1, 2, 3, 4, 5, these top 5 genera were all lumped in one genus Neramycia. Yet they are very distinct genetically, and I'll show in a moment they're also distinct morphologically. And by having them all combined, it makes it difficult for us to see exactly what those relationships are. If you just lump everything into one, you're not going to be able to see. I think somebody earlier was talking about granularity. You miss the details in this. So those two genera existed before, Leiphotis and Nycticenops. So we elevated those genera. Neramycia we confined to a small group over here, but then we introduced 2 new genera, Afronycterus and Pseudoromycia, to fill in the species in between. And it's caused a lot of frustration by people. People still come to me and say, why did you have to give us new names? But this is the reason, because those are real groups, and they didn't have any names before. And this is just to show morphologically what I said, that morphologically they're very different. This is taken from this is just one aspect of morphology. It's the baculum bone. All bats have a little bone that sits in the penis, at the tip of the penis, that's important for reproduction. So having differences in the baculum has implications for reproduction. And of course, reproduction is vitally important for speciation. Look at those four bacula. Again, you don't need any specialist training to see that this baculum here is totally different from that one, and this one, and that one over there. So these are clear genetic groups with clear morphological differences, but it had been overlooked because no one had looked at this aspect of morphology. Their skulls are very similar, and not a useful tool in this particular instance for identifying genera. Now, taxonomy often requires collaboration, because it's not possible to do this on your own. The only reason I was able to work on this is because I was given the opportunity to work at the Field Museum. And ironically, the Field Museum, which is in Chicago, which is in the USA, which is about as far away from Africa as you can get, it has one of the best collections of bats in the world from Africa. And not only that, but they had sequenced all of those specimens that I worked on. They had 1,200 specimens of vespers in their collection that had been sequenced and for which there was morphological, well, there was a specimen there. And there is no place in Africa that we can find a similar collection. And that is not right. We should have those collections here. Our own governments should be investing in this, and we should be developing the resources for us to be able to do this work. And we all know the frustrations of travel. For us to get out of the continent, we need visas, and the visas require this and that and the other, not to even mention the expense. But on the positive side, it got me to collaborate with all sorts of interesting people. That is Paul Webala here. He's a very well-respected Kenyan bat biologist and very important player in this particular paper. That's Terry Dimas. He did all the genetic work. And this was Bruce Patterson, who hosted me at the Field Museum, and plus many other people as well. So someone earlier, not someone, but Kendra talked about networking, and this is like a mini kind of networking that I had to do, and a very fruitful one. So this is the last thing I wanted to show, is that taxonomy can also be vitally important for conservation. This is something that a lot of people don't realize, but conservation areas of conservation importance are usually based on two things. One is that it's areas that have high diversity of species, especially unique species. And the second is that conservation efforts really are focused very often on endemic species. But those endemic species and those areas of high species diversity, you need taxonomists to be able to tell you correct species identifications, because if you have misidentified species or if you haven't identified them correctly, you think that an area has lower species diversity than it really does. When I started working at Mount Nimba, there were 32 species known from the mountain. By the time I finished, we are now at 59 species. And I know that there's a 60th species because I saw it, but I couldn't catch it. And I don't know what it is, but I know it's not a species that I already have. So it's doubled the species diversity. And that makes an enormous difference in terms of how we see it from a conservation perspective. Here's an example from Southern Africa. That's the distribution of Miniopterus mozambicus. Forget about those, but in the blue circles, there's a second species, Miniopterus wilsoni, that we recently described, and I just realized, my apologies, I updated this on my point. I didn't save it over here. So this is Miniopterus wilsoni, the same Miniopterus that I showed a bit earlier in this earlier slide. So this area here, where these dots are, there's two species of Miniopterus endemic to that area, Miniopterus mozambicus, Miniopterus wilsoni. Before I did this work, we didn't realize that there was any endemic Miniopterus in this area, because all of this, it was just simply called Miniopterus natalensis. So how can you identify this area as an important biogeographic area if you think that the species there are just common and widespread to the region? Basically, you can't. So there's been intensive work that's been done in this area of northern Mozambique, spearheaded by Julian Bayliss over here. and I've joined him in the field for a number of his expeditions, and we've shown that the fauna that's associated with these inselbergs in northern Mozambique is unique and doesn't occur elsewhere in the continent. And the species there that were originally named, they were named incorrectly because people just lumped them in with the closest species from other parts of Africa. So this entire area was overlooked as an area of high endemicity, an area of high endemic species, in other words, species that don't occur anywhere else in the world, simply because we had not done the taxonomy correctly. So taxonomy is also vitally important for conservation. This book was published in October 2020, so it is very much up to date, but just to show you how quickly the field of taxonomy goes, all the things I talked about here with the Vesper taxonomy updates is not included in this book because that stuff was published in 2021 after this book was, after we had done the revisions for this book. And unfortunately, we weren't able to update the taxonomy for some of the bats here. And I like to always end off with this slide. It's a slide taken by Peter Naskreki. He photographs, but he's from Mozambique, by the way. I know there's two colleagues here from Mozambique. He lives in Chitengo in Gorongosa National Park, and he photographs bats in the wild, free-flying bats in the wild. And if you look at these photos of bats, we rarely see bats in this light. Usually we see them either as museum specimens or someone holding them, but look at how interesting these guys are in mid-flight. Okay, thank you very much.